Hi, everyone. We'll give everyone a few minutes to sign on and we'll get started with the webinar. All right, let's get started. Thank you everyone for taking the time to join us today. Welcome to this webinar, the usage of online monitoring to determine derouging and passivation frequency. My name is Ernst Kornpan. I'm the product manager at Cosasco, and I will be your moderator today. A little bit of housekeeping before we get started. The webinar today will run about 1 hour with 40 minutes of presentation and Q&A at the end. If you have any questions or comments during the presentation, please feel free to type this in the Q&A box below. And if by any chance you missed on some important points, we'll be recording this webinar and we'll be sharing the presentation with all the attendees right after. So now let's get started. I'm very excited to have our speaker today, Nissan Cohen. Nissan is a recognized worldwide expert with over 40 years of experience in high purity, ultra pure, reclaim and recycle water systems with profound expertise in instrumentation, automation, and organic contamination oxidation system using ozone, UV, ion exchange, and catalyst. He is an accomplished author of over 40 technical articles and major industry and regulatory publications published by the International Society for Pharmaceutical Engineering, ISPE, ASTM, and the Journal of the Institute of the Environmental Science and Technology, to name a few. And now, without further ado, I'll turn it over to you, Nissan.
Thank you very much, Ern. Good morning, good afternoon, or good evening, depending upon from which area of the world you are watching this particular webinar. We should thank you for your participation, and we will be talking about science-based instrumentation in order to determine derouging and passivation frequency. For today's agenda, we're going to talk about certain different areas having to do, number one, with the background of Rouge and its development, the technology of corrosion monitoring, the applications of Rouge monitoring in pharmaceutical water systems, and why we do that. There will be an extensive area on data examples, charts, and graphs showing real data from real applications that have been amassed throughout the world. We're also going to have a section on comparative Rouge tests and observations, which means there are different ways that you can actually measure Rouge, and we will show the viability of both. And then there'll be a conclusion slide before we go into Q&A at the end. Background, let's talk about where Rouge comes from. Rouge is a thin film iron oxide or hydroxide, usually reddish brown or golden in color, could be other colors also, and it's typically found on stainless steels, but Rouge actually develops on all different kinds of metals. All metals will rouge over time, depending upon their interaction with hydroxyls. Rouge can also consist of iron, chromium, nickel, and tungsten oxides in stainless steel because these are all part of the constituent materials of 316L stainless steel. Tungsten, believe it or not, is actually more readily available in a lot of newer stainless steels because stainless steel, about 75% of all the metal in the world, is recycled stainless steel. And when you recycle stainless steel, you use tungsten. So besides the traditional iron oxide and chromium oxide, nickel oxides, molybdenum oxide, you can also have tungsten oxides in your material for Rouge. Rouge is prevalent in biopharmaceutical applications because we use a tremendous amount of stainless steel, primarily 316L, but other stainless steels can be used also, like 304 and other more exotic alloys. The point being is that rouge will develop and is more prevalent in higher temperatures. The higher the temperature, the more rouge is actually developed, and we'll show reasons for that shortly. Rouge normally forms in the interior surfaces of high purity water systems, storage vessels, and distribution systems, including piping, valves, pump housings, pumping themselves and impellers, fittings, and the process vessels. So anything that comes in contact with the water will rouge. Depending upon the temperature of the water and the usage and flow, will also determine how much rouge develops over time. There are three different classifications of rouge. First classification we call class 1 is what we call non-adherent migratory rouge, and it does not affect the or interact with the actual surface. It can be easily removed from wipes. Traditionally, when we see rouge on pump impellers, etc., that rouge is a very fine dust particulate, usually between 20 to 50 microns. And those small little particulates are non-adherent. They do not interact with the surface and they can be wiped off readily. That's class 1. Class 2 rouge is a little bit different in that it causes pitting and corrosion and that beautiful patina that you normally see on interior to different pieces of piping, vessels, and different pieces of equipment. It can only be removed by chemical etching. You cannot remove it with a wipe, and it normally forms what we call hematite, which is a more complex ferric oxide. Class 3 rouge, which is normally blue, black, or dark purple, is somewhat benign. It actually formulates almost an annealing effect and a coating onto the piping itself. It can only be etched off of the surface with using phosphoric acid at about 80%. It rapidly reforms even after you clean it off. It is normally found in steam systems, high purity steam systems, pure steam systems, etc., or in very high level water systems, which may be running above 90 degrees Celsius as an example. So you can find it there too. This particular type of rouge is actually magnetite, which is a more complex ferric oxide than even the hematite or ferrous oxide is normally. So here are a couple of examples of rouge. Over here, we're looking at a pump impeller and a pump housing, both on the top and on the bottom. You notice there are different colors, different hues, depending upon the temperature and the velocities. The bottom unit are primarily non-adherent rouge, meaning rouge develops and then will migrate throughout the system due to the impellers, and the pump themselves can be removed with wipes. Some of the residual rouge that might be at the bottom can also be non-adherent, at the top you'll see is more of the rouge that you would find for class 2 rouge. Bottom would be class 1. Here's an inside of a bore where you get to see those very different colors of rouge, from goldens to different browns to reds to deep brick reds throughout the systems. And these formulate due to the temperature and the exposure from the water. Both of these rouges, this rouge over here is the corrosion, showing the actual corrosion. Corrosion means that there's metal loss. And sometimes there's metal loss, sometimes there's an annealing effect. This is that blue-black rouge that we talked about earlier, or purple. This is actually a spray ball. And you can see the color and that the rouge is actually an annealing effect. It's almost coated it completely. That rouge will not fall off. It will not come off. It can only be etched off with chemical etching. There was a bag put around this because of QC/QA interference with engineering. They felt that this could cause some downstream problems, so they asked engineering to put a bag around this particular spray ball. Of course, if you know what magnetite does, it will not come off of this. There's no possibility of flaking. Unfortunately, sometimes QC/QA people determine that they are more knowledgeable about rouge than the engineering, and then you can have superfluous issues like this, where a bag is put around a device in order to stop particulates, It will actually never happen. All these different rouges are corrosion. This one over here in particular, as you can see from a WFI water tank at the top and a vessel, WFI water vessel with a spray ball in the bottom, the rouges can get very extensive over time, and the color itself can be very, very intense. The point is that color is not necessarily indicative of the amount of rouge that could possibly flake off or get into your product. So having color and looking at color without a determination of rouge development and the depth of rouge is there is not necessarily indicative of when the frequency of your rouge monitor should, is not indicative of when you should perform a derouging or passivation. So color is subjective. It is not necessarily indicative of particulates that may be in your water. Sometimes there are filters that are on water systems. You can see this filter over here has a slight pink hue to it. That pink color is the actual stoppage of those non-adherent rouge particulates that were coming off of the pump system and stopping it from getting downstream. Most WFI water systems, due to a previous 30 to 35-year-old trend, have not put filters into their system because of a FDA derivative guidance that came out 35 years ago that said you can't put filters on to control microbials. So they decided that you couldn't put filters anywhere on a WFI water system. That's not true. You can, especially for particulates. Point is that what you're looking at here is deposition. What you were looking at previously is corrosion. So there's a difference between corrosion and deposition. Deposition means that all different pieces of plastic and non-metallics in water systems, including gaskets like ETFE, PTFE, etc., have a slight negative charge. And that slight negative charge will adhere and attract positive charged particulates, in this case being iron oxides and ferric oxides. So what happens is, what most people don't know is that these non-metallic gaskets and materials can attract rouge, which is a good thing because you're stopping the rouge particulates from cascading down into the water system, down to the points of use. Many drug companies are concerned about the possibility of particulate matter contaminating their products in sterile systems. Effective way to maybe to deal with this problem would be to monitor the rouge rate online in order to prevent process upsets and prevent contamination downstream. If rouge does affect the product, though, the product is deemed unusable because of particulate contamination. You might be familiar with one of the newer guidances that came out from the USP talking about elemental particulate contamination in products like APIs, etc. Rouge rate development is currently unknown, and rouge formulation rate is currently unknown. So unfortunately, we don't know how these particular rouge effects actually develop and what their formulation is. Every different water system can be different due to the materials of construction and the amount of water and temperatures that are used at any given moment. Since the determination for passivation frequency and the depth of rouge in order to do the passivation frequency is unknown, there were previous attempts that were using coupon systems that allowed coupons to be used in order to determine rouge frequency. Those coupons would subsequently be removed and then sent to a laboratory and see how much the rouge depth there actually was. Unfortunately, you had to shut down the water system in order to extract these coupons. And basically, when they went to the laboratory and they came back with a number, everyone kind of said, okay, what do we do now? So in order to get away, the coupon systems were given up. And today, most of the determination for derouging passivation is determined on discoloration or a time interval determined by QC queasiness. QC queasiness is the QC department's tolerable level of what they think is doable until you can actually do a derouging passivation. It's really based upon a subjective determination on their part of what they think that frequency needs to be. So it's not determined on science, it's determined on their gut feeling of what's tolerable or not. The industry needs a method to determine the Rouge formulation rates and determine the Rouge thickness as it accumulates over time due to the corrosion aspects. And we also need to be able to have a scientific method to determine when the frequency should be performed for both passivation and derouging. This means that all different types of water systems will rouge at different rates depending upon their temperature, their operation capabilities, and how the structure of the system is actually determined. An online method which integrates with the existing instrumentation, network, data logging, and computer systems is extremely important. Almost everything we have today on modern water systems are automated, and we should be able to have a system that integrates with that automation and computer system also. Something that should be easy to calibrate and easy to maintain is always needed in our industry. So if you have any other questions before we go into the next section, please write them down in the chat box so that we can deal with those questions later. When we're looking at Rouge monitoring, Rouge is a form of corrosion, and that corrosion is the electrochemical phenomena based upon electron migration with a combination of a hydroxyl radical. Whether you're oxidizing organics or oxidizing metal, it's both the same way. And to prove that corrosion is a migration of electrons and the loss of metal, think about the Titanic sitting at the bottom of the Atlantic Ocean. It's not gaining any more metal. It is only losing metal. And over tens of thousands of years, I'm sure that the Titanic will actually dissipate into the water it surrounds it. Point is, corrosion is metal loss. It is not a deposition. Corrosion, though, can be determined by measuring current across a potential anode and cathode, which is what a Rouge monitor actually does. Over here, what you're looking at is an equation at the top showing iron, water, and oxygen, which is going to yield a certain amount of ferric ions, which combines with the hydroxyls in order to formulate ferric hydroxide. You notice at the bottom, you'll see an anodic reaction and cathodic reaction. The anodic reaction actually is where the iron is liberated from the metal. What happens is as the iron is liberated from the metal, electrons migrate over to the cathodic side, while the iron combines with the hydroxyl radicals, formulating the ferric hydroxide, And the electrons will formulate with the water and oxygen more hydroxyl radicals. The more hydroxyl radicals there are in the system, more liberation of iron there's going to be as the corrosion develops on the anodic side of the metal. This is a graph of temperature showing that the hydroxyl rate will increase with the temperature over time. And the oxygen content will be degasified nominally due to the increased temperature. So as the oxygen and the gas of the oxygen decreases due to temperature, the hydroxyl rate actually increases. This next graph, though, actually shows the two different phenomena that are apparent in the water system. One is the chromium oxide development rate, which is the passivation layer that's there in order to help prevent a certain amount of oxidation. Chromium oxide, by its nature, only develops as a layer at about 20 to 25 angstroms, or 2 to 2.5 nanometers thick. That's all the layer is. This is not a firewall that prevents corrosion by having passivation. So the chromium oxide layer is very, very small. The ferric oxide, though, as you can see here as hematite, actually develops more strenuously as the temperature increases. The higher the temperature, when you're running your system at 80 to 85 degrees Celsius, will cause the ferric oxide rate to develop very highly. This is the Stern-Gary equation that was developed in 1957 in order to show what happens with corrosion. What we're looking at is the probes down here have an anodic and a cathodic aspect to it. So that anode and cathode, and you're allowed, you measure across the eye measure the actual rouge development over time. So as the corrosion shows what the corrosion is between those particular poles, you can then calculate what the actual corrosion development is by using this equation. What this allows us to do is each of the probes is manufactured out of 316L stainless steel in order to exactly emulate the 316L material in your water system. The idea is once the probes are installed in the water system, they are not removed. And the reason that they are not removed is because we want the probes to see the exact same chemistry as the water system, the piping, the vessels, and the pump impellers that are in your system. That way it's very indicative of how the probes see the water chemistry as do the piping and vessels. We want it to emulate exactly of what's transpiring in your water system. So what are some applications? Well, the idea here is when to perform derouging passivation. Derouging passivation often is a minimum of a 3 to 4 day downtime and longer for its application, depending upon how large your water systems are, how many loops are in your water systems, and whether you're passivating both your purified water system and your WFI water systems as an example. Don't forget, purified water systems that are using heat sanitization are often run at heat temperatures, elevated heat temperatures often. So mainly you're looking at derouging passivation of both your PW systems and your WFI water systems over time. There are chemical costs associated with doing derouging and passivation. Primarily, we use citric acid in the United States, but nitric acid and phosphoric acids are very prevalent, especially when there are large amounts of derouging and passivation to be administered. But when you add acids to the system, you have to have acid waste neutralization by adding caustics in order to countermand the pH that is due to the acids and bring it back into around 7. So we add typically sodium hydroxide or ammonium hydroxide in order to countermand the acids that were applied in order to do the derouging. You have labor costs in manpower, and you also have production shutdown. Because you have shut down the water system in order to perform derouging passivation, you cannot produce product during the same amount of time. That loss of production means that there is a certain amount of costs associated with the loss of production at that particular time. And the frequency, how often do you perform this particular administration? Is it every 6 months, once a quarter, annually, every 18 months, biannually? And how is that determined? If you determine it only by the color of the vessel, you may be derouging and passivating on a much higher frequency that is needed, which means that there is a considerable amount of cost in chemicals and wastage and water that's being used that doesn't have to be, because you are performing the derouging passivation at a much higher frequency than is needed. This is a typical water system we see in a pharmaceutical application, showing a purified water at the top and in water loop, and then running a multi-effect distillation unit as an example off of the purified water, then running into a WFI water storage tank, which may be running at hot at 80 degrees Celsius with a pump downstream, and then a recirculating loop back to that hot water storage. The point is that both the purified water loop and the hot water loop, especially if the purified water loop is heated for a hot water sanitization, are susceptible to rouge, and both will need derouging and passivation over time. The costs of derouging and passivation for a small system are being elucidated in order to show even a small system what the actual costs are. This showing a 76 per liter or 20 gallon per minute system of USP purified water running into a 4-gallon or 15-liter per minute distillation unit, which is a small multi-effect distillation unit, running into a 38-liter per minute or 10-gallon per minute WFI storage and distribution, running with a 2-inch piping, about 300 meters or 1,000 feet of piping through the distribution system and returning back over to that storage unit. Even this very small system, though, in chemical costs and labor alone to do the administration for derouging and passivation will cost at least $30,000. If you look in the downtime of production in days, the client's labor, the non-production of products over the time period, the expenditure and the cost of the system gets very, very large. Here showing only $50,000. If we look at a normal, fairly decent sized production facility running vapor compression distillation units at 3,000 gallons per minute per hour, as an example, feeding a couple of loops, that particular type of a system will cost about $275,000, almost $300,000 per administration for derouging and passivation, etc. So these numbers can get very, very large, very, very rapidly. If you don't do a derouging passivation once every 3 to 4 years because you have an online Rouge monitor which can tell you what's going on, you can save hundreds of thousands of dollars to millions of dollars for the company and over a couple $100,000 or greater per site. So that's a lot of money in order to be reconciled. The configuration of what we supply actually is a Rouge monitor, which is a wireless Rouge monitor. That wireless Rouge monitor has a probe that goes inside the water, has a clamp to your extension for your T that would normally be 316L. And then you have an adapter that runs on that particular unit up to the actual wireless unit itself that does the integration of the data that's coming in from the probe. Everything is run off of a battery that's sitting inside the top part where the radio and the wireless unit then communicates to a gateway. That gateway actually allows it to have direct communication to all of these units that might be out in the field. And that runs at 2.4 gigahertz on a random radio basis. It will integrate the data coming in from the wireless unit that's out in the field and then send that information over to a computer system, whether the computer is standalone or maybe a SCADA system, a Delta V or whatever. There can also be Modbus TCP IP communication between the gateway and the particular unit. The batteries that are inside the wireless unit will last for a 3-year duration under normal usage. So it's a fairly easy integratable system. There are no wires to run. We don't have to worry about additional electricians, etc. There is a small adapter, a converter that runs into the gateway in order to provide 24-volt VDC to that particular unit. Very interesting that we can add and subtract units throughout the configuration with very little integration. Where do we put these particular units? Well, on our hot water WFI water system, the best place to put them is directly after the pump. Usually there are 2 pumps. One is on standby or an auxiliary pump, and the pumps are interleaved on a separate basis in order to have both duty cycles for both pumps. After that pump, where there might be a T, would be a good place for the installation of a single wireless unit on a single system. The reason being is that the pump has very high cavitation, high velocity, and that 2.5 nanometer passivation layer is eroded very, very rapidly directly after a pump. Inside the pump itself on the impellers, you cannot obviously put a monitor there, but the monitor is there directly after the pumps because the pumps will generate the greatest amount of rouge as far as particulate rouge is concerned. The unit actually doesn't pick up the particulates themselves. It actually picks up the amount of rouge and how the water, the interaction of the water with the piping in the vessels. It may be a very good practice, though, to also have a second wireless rouge monitor on the return line going back into the storage tank. That water shows the nominal aspects of what the water system and water chemistry and temperature of the water system is doing at any given time to the piping and out to the points of use. So having those two units is a good comparison because you have a fail-safe situation where if one unit should fail, the second one is operational. You can also look at the comparative data between the two different rouge monitors online. Depending upon how many loops you might have, it would determine how many wireless units might be in the whole building. You still only need one gateway, as that one gateway can accommodate up to 100 actually wireless units out in the field. We also have an application for steam and steam condensate. If you look at this particular drawing, at the top part of it over here is a Spirex Sarco unit that allows you to have steam condensate. Up here on the top part will be a line for where you're running your steam with a 1-inch flange coming off of that steam system. That will allow the steam condensate unit, the Spirex unit, to convert the steam into water, and that water will then drip down into this fitting that we have where the actual probe is installed at a 180-degree angle or a 6 o'clock position. The idea is that there are two valves. The bottom valve has a simple ball valve on it in order to shut it off, in order to create a well, and that well builds up with water, and the top valve over on the left-hand side will be going to drain. So this is the water it's going to drain. The probe will be measuring the amount of rouge that develops based upon the temperature of the water. Even though this is not a singular steam application, it is very indicative of the amount of rouge that's currently developing in the system and can be used for derouging passivation frequency for your steam system. We're going to go through some data examples, charts, and graphs. You can still put your questions together through the chat box if you'd like for any given section. This is a graph showing company one and showing the beginning of the day the rouge monitor was installed. Red always represents in these graphs the actual totalization of rouge over time, and the blue line actually represents the amount of rouge that comes out every 10 minutes in looking at rouge status, rouge development due to operational characteristics of the water system. It takes about 24 hours for the rouge to actually get into the system, the rouge monitor to stabilize inside the system in order to show its development. You look at the screen over here on the top side, you'll notice it's running at a half a nanometer, 1 nanometer, up to 2.5 nanometers on the rouge development. On the bottom screen, it's running from 0 to 50. This is the same site that we're looking at. Again, the January 14th for the date of the rouge monitor installation. And this shows what happened over the next 2 months, where the red line shows the totalization of rouge about 7 nanometers. But the bottom line, if you look at, shows the status of the rouge at any given time due to operational characteristics of the water system, what's operational, what's recirculating, the temperature of the water, and other operational conditions. What we're going to look at specifically is this section over here between the 18th of February through the 26th of February in order to look and see what the water system was doing. Notice that again, the totalization is only 7 nanometers over those 2 months. So here's what's transpiring. If we look at the left side of the graph, we can see that there are units that go up, and then there's values that go down. The values go up, the values go down. This is actually based upon the heat exchanger in the water system. As the water is recirculating without any usage in the system, the temperature goes up and down and fluctuates due to the heat exchanger. When the water goes down to 75 degrees C, the heat exchanger comes on, and there's a subsequent increase in rouge development. When the water goes down, when the temperature goes down, and you'll see that there's a decrease in the amount of rouge development. But because this oscillates up and down, you see that it actually stabilizes over time. It's running very, very normal. But at this particular point, you can see that there's an actual drop. And what happened over here is the water at the point of use was being used, whether for filling or for actual operation. And you can see that the rouge development drops precipitously over that time where the water is being used. Why? Because the water that was recirculating had a more contact time with the actual vessels themselves in the piping. When the point of use is open, there's a less amount of residence time of the water in the system, which means that there's less amount of rouge being developed in the piping itself. You can see over here that the rouge goes up and goes down. These were actually the sample time was the sample was opened up for sampling points and then shut off. So there was a certain increase in rouge for sampling and sampling of the system at the points of use. Over here at the bottom part, you can see where there was filling operations once again. And then again, over here where the filling operations were shut off and the water system started to increase rouge because of the contact time and the heat elements associated in the system. If you look at this over on a developmental story basis, the red line over here represents the amount of time it takes you, the amount of kilometers or mileage that it takes from you to get to an airport. This line over here on the operational, which is the blue line, is determined about, as an example, on the freeway, how much traffic there is, whether there are traffic stoppages, whether you use your braking and you're accelerating, you're braking and accelerating. It's the operational characteristics of your car. We both agree that it's a certain amount of miles in order to get there, but the actual amount of usage of miles per gallon is going to change dramatically due to the influence of how the car is being operational. This is the same system, but another loop running at 50 degrees nominally and taking it up every night to about 80 degrees Celsius. You can see this 80 degrees Celsius every night going up on the bottom line, on the bottom graph, showing these small little peaks going up and down. But the actual increase over the time of 2 months is only about 1.5 nanometers. We look at this data on a more concerted basis. You'll see that the heat exchanger comes on at 7 o'clock at night. There's an increase until 8 o'clock, to 9 o'clock, where it's run for 80 degrees Celsius. From the 50 degrees Celsius is then cooled down and the rouge rate goes down, but over time. However, if you look at the red line, you'll see that there's virtually no increase whatsoever. This is the same site. This is the day on August the 21st when they did a derouging function of using phosphoric acid at 80%. And then over here, you'll see on September the 8th, downstream, there's a slight increase on the top line, showing that this is when the passivation was actually incurred. So there's derouging on the 21st, passivation on the 8th, and here's what transpired over that time. At 4 o'clock in the afternoon, the phosphoric acid was administered. During that time period, when the phosphoric was administered, you'll see that there's an increase in rouge. Why? Because there's more, we're reducing the metal on the piping and the vessels, and that increase in metal is shown up in the rouge. The chemistry, again, against the probe, shows exactly what's going on with the metal itself. So after 3 hours, which is a normal practice of most third-party rouge chemical administrations, they come on and they say, okay, 3 hours is up, and at 7 o'clock or 1900 hours, they administered sodium hydroxide. But what happens over here is you can see that there's actually still a large increase in the derouging, and the Rouge development and the corrosion due to the chemicals being administered. And even after 3 hours that they administered the sodium hydroxide, there was still corrosion developing. It actually took 22 hours for this system to come back under, to stabilize back as it was previous to the administration of the phosphoric acid. This is really important because the vendor would tell you that everything happens after 3 hours and your water system is just fine while they're rinsing down. But if you use conductivity only as an assessment and not a Rouge monitor, you really don't see that your system comes back under compliance for 22 hours. That's really important. The derouging, the passivation chemical that was used at this particular site is something called Direnutra. Direnutra running at a pH of 7 didn't have a very big influence on the increase in nanometers. You can see a slight increase over here on the top. If you had used citric acid as an example, instead of the Direnutra, because it has a more acidic level as an acid, you would have seen more of a spike that was shown over here as you did in the derouging in order to do the passivation. Data is in single-digit nanogram levels. So we're between 15 to 40 angstroms, very, very small amounts. That's what the normal range. The real-time changes in the Rouge rate, though, are due to point of view usage or not, flow rates, temperature fluctuations, and operational characteristics of your water system. Temperature fluctuations also taking it up and down. To understand if Rouge is in the product, though, you need to be able to measure the product at very small levels, primarily at PPT levels, parts per trillion. The accumulation rates, though, over time are easily interpreted because we have accumulation in Rouge directly on our Rouge monitors. And we can see what the Rouge development is due to periodic development, due to temperatures and operational characteristics. We can set alarm limits, both specification alert, action, and action alert and specification limits on our systems, on our computers, in order to see what's going on with our water systems. Those alarm limits can be set for very low levels. And we can also determine what the frequency is for derouging passivation upon real data. So we have a scientific assessment for derouging and passivation. Go over to the next section on the comparison. We'll actually be able to look at the data in order to determine what those ideas of development should be. So you can use Auger electron spectroscopy, which is a destructive way in order to look at derouging deposition development over time. And you can take a piece out of spool piece, send it off to your laboratory, have the laboratory do x-rays, and they can tell you what the depth of the Rouge is. What we did is we actually took Auger electron spectroscopy and compared it against an online Rouge monitor for the same system over a period of time. And that would show that the values are on the next slide. We also wanted to take a look at if there were investigative variances due to water classification temperatures that would cause any variances between an online Rouge monitor and the Auger methodology. So here are three different companies, company A, B, and C. Company A had a Rouge monitor on there which showed 70 or 700 nanometers of development of Rouge over an 11-month time period. The Auger electron spectroscopy also showed 750 nanometers or 0.75 micron development. So there's very good readings between the two units. This particular water system was abominable. It had what we think were components that were 304 stainless steel, might have even been some other steel that was in there that caused the derouging and passivation to move up very, very rapidly. But the agreement between the Auger and the Rouge monitor were very, very close, within 50 nanometers and about 6% deviation between the two difference. Company B, which was a more modern water system, fairly brand new, and you can see there that the Rouge rate development was only 75 nanometers over 10 months. And the Auger showed that it was 80. Again, a very good agreement between the two at 5 nanometers. Company C was an older system, about 12 years old, had been derouged and passivated annually for the previous 12 years, had a slightly larger increase in Rouge reading per year, running at about 170 nanometers, with the Oxide showing 160. Again, very good agreement between the two of them. The point is, now that I have the data that shows what my Rouge rate is over time, I can now estimate when my derouging passivation should occur. You know, a half of a micron is a very small amount of Rouge. That's less than a half of a micron. And you know, 500 nanometers on company B as an example might be 5 to 6 years in order to attain that level before doing a derouging passivation. Even company C, if I set it up at 400 or 500 nanometers, maybe 2 to 3 years in order to accommodate that. The point is that I can set up my Rouge derouging and passivation frequency based upon real data. And real data means that I will not be performing derouging passivation based upon an annual basis or every 18 months, but based upon real data of when I achieve a certain level of an action limit, an alert limit, long before a specification limit that might be determined by a URS. Couple of observations about this particular system. Operating temperatures, ramp-up temperatures, pharmaceutical water classifications had no influence on the comparability of the two different methods. However, the differentials that we saw were very, very small. And the variances and percentages were extremely small also. So we could actually use Auger electron spectroscopy as an independent method to verify the accuracy of an online Rouge monitor, or we can use 2 online Rouge monitors on the same system in order to show comparative data, much like we show with TOC analyzers. Couple of quick conclusions. Online determination of Rouge development, its thickness over time, without sampling, without labor, without laboratory testing, is inexpensive, it's dynamic and integratable to existing computer and networking schemes. Passivation, repassivation frequencies should be based upon science and need, and not QC queasiness, not color, not because I want it done once a year, not because we do it we do everything else once a year. It should be based upon real science. Rouge monitoring units are inexpensive. Saving 1 passivation annually will pay for the unit and the installation many times over. Depending upon how many loops you have and how large your water system is, the savings could be hundreds of thousands of dollars to $750,000 or greater per site. And if you're looking at company-wide of setting up a Rouge monitoring system on a multinational, the savings are millions of dollars per year. Installation of the units, though, should be based upon the amount of loops that you have and doing a risk-based assessment of your system. So now we're going to turn it over to Ern, who's going to ask us your questions, hopefully, and we can move on from there.
Thank you very much, Nissan. And just a friendly reminder to everyone, if you have any questions, please feel free to type in the chat or the Q&A below. So Nissan, one question coming in, what are the NACE standards that apply to this Rouge corrosion?
There are no standards per se, except it is stated by the FDA and by others, other regulatory entities, that if you have Rouge, you need to have a mitigation system and a plan of how to deal with the Rouge. Rouge over time, if it is untouched and not mitigated, will cause corrosion of the piping that will lead to pinholes in the piping and the vessels that will cause problems and deformations over time. So Rouge needs to be addressed, but there is nothing in the United States Pharmacopoeia or any pharmacopoeia that states that you need to monitor Rouge as far as water quality is concerned. You can have water systems that don't have Rouge. You can have water systems that are made out of PVDF and run at ambient temperatures. You can have water systems that run at 4 degrees Celsius. So temperature of the water is not a criteria for either WFI or purified water. But Rouge will develop more succinctly in hot water and needs to have a mitigation plan.
Absolutely. Thanks for that, Nissan. And the second question is, just to clarify, does the Rouge monitor measure corrosion directly, or does it measure adherent Rouge? Can it differentiate between Rouge classes?
Can it differentiate between Rouge classes? The answer is no, it cannot. It will measure the amount of Rouge that develops as far as the actual Rouge on the vessels and the piping. The particulates that are developed inside a non-adherent Rouge are a specific aspect of how that pump operates and the impellers themselves. The non-adherent Rouge is picked up marginally by the Rouge monitor itself. It's more the interaction of the water and the chemistry with the piping and the vessels, which causes the hematite, again, the hematite and the magnetite. Don't forget, we do not do derouging for particulates. We do derouging in order to get rid of the corrosion that's actually on the vessels and the piping. Particulates can be removed if it's a problem by filtration, and that's the easiest way in order to get rid of them.
Thanks for that. How do we determine the derouging frequency then based on these monitoring system, and how do we set limits?
It should be based upon a equally devised level of value between you and your QA/QC department. As an example, once we said again, the color of the Rouge does not make any difference. It's actually what happens over time. Again, Rouge can be developed very slowly or fairly quickly, depending upon the components that are in your water system, the temperatures that you run at, whether you're running at 80 or 85 degrees C continuously, etc. So not all water systems are going to Rouge at the same rate, but you perform your derouging passivation by gauging a value that you want to determine. If I determine that 400 nanometers is the value that I want to do my derouging passivation, and I'm only running at 60 or 70 nanometers per year, it may take me 5-6 years in order to get to that level before I do a derouging passivation. That's quite a possibility. The point is that derouging and passivation should be based upon science and not based upon color or QC queasiness. So you can determine whatever that level is. Traditionally, when I've done these presentations before, we talk about 400 nanometers being probably a very good level. It's way below 1 micron. It's below a half of a micron, and it's achievable over time. But you can set it up for whatever you think, whatever value you think is indicative of your water system, whether it be 400 nanometers, 500 nanometers, 300 nanometers. It could be even higher, as high as 1 micron, but it needs to be a number that is going to be assignable both by you and your QA/QC department.
I saw that we have another question coming in about the limit as well, but I hope Nissan was able to answer that question already. If not, please let me know. The next question is, are you finding many companies that are using this kind of methodology to time their derouge procedures? It seems that most either do it based on schedule or problems, like you mentioned, Nissan.
Hey, you can do it by schedule as much as you want to. How much money are you wasting by doing that? So my comment to you is, do you really want to perform this as an annual basis where your URS was set up and your SOP was set up probably 10, 15, or 20 years ago based upon not being able to have a Rouge monitor at that particular time? Or do you want to bring yourself into the 21st century where you can actually do derouging or passivation based upon real science and integrating that particular monitor into your computerized system that you currently have? Sure, if you want to go out and spend money on do this on a periodic basis without any Rouge monitoring, go ahead and do that. However, if you want to look at the economics and being green and how much money is actually costing your company, you may be costing your company company-wide millions of dollars a year in a performance of an administration of chemicals and a process that you do not need. How do you justify that?
Thanks very much, Nissan. Another question is, are the sensors and transmitter 3A or HEDG approved? Also, what is the IP rating of the transmitter? Is it IP 68 or 69?
I think I do not know what the rating is per se. They're not explosion proof. And I think that they were originally IP4, I think is what the rating was.
Yeah, and I think to follow up on that question as well, we'll definitely send out more information after the webinar that details out all these technical specifications as well. So moving forward, what's the usual pressure for this kind of system and the pressure rating for the clamp that you mentioned during the presentation?
The pressure is your regular pressure rating that you have. Actually, the systems can go up to a considerable amount of pressure because they were designed originally for oil and gas, where the pressures are much higher. I think up to 200 PSI as an example. So there's no issue with the units themselves as far as the pressure in our normal operating systems.
Absolutely. So I believe this is a follow-up to the previous question on the NATE standard. If there is no standard, what would a choice be from the client once this is not forced to apply any monitoring system? Is there any recommended practice from any entity that we can follow?
I think I already addressed that. The point is that the issue is you have to have a mitigation plan as far as the regulators are concerned in order to deal with Rouge. There are no standards worldwide that are stated for Rouge because it's not a water quality issue. It's an operational issue of your individual water system and at what temperatures you're operating your water system at currently. So the point is that, again, since there is no worldwide standard, you have to develop up limits that are based upon action, alert, and specification limits based upon a URS that you might have for your own system. So how you establish that is to state unequivocally that Rouge is prevalent in hot water systems. We have hot water systems. We need a mitigation plan. We also want to do the mitigation plan based upon science and technology of having an online Rouge monitor and not doing it based upon subjective issues of color, discoloration, or QA/QC saying we do everything else once a year, we're going to do this once a year.
Absolutely. So can you share a bit more whether this monitoring system needs any calibration, how it's done, and then what's the recommended frequency?
Yes, each individual system can come with a calibrated test probe. That calibrated test probe allows you to have a level of connectivity between the probes in order to show that the probes are operational. The calibration verification probe can be installed on the wireless unit by removing the adapter that is between the flange. So the flange remains intact with the actual probe, and above that there's a small adapter. That adapter is taken out, and the test probe is placed into the bottom of the wireless unit in order to test doing a test verification. So when you want to do a calibration verification once every 6 months or once a year, you do not have to take the probe out of the system. You do not have to take the water system down. You simply take the adapter off, put the test probe into the unit, and that will tell you that the unit is under calibration verification.
Absolutely. So we'll probably be able to take about 2 more questions. One being, for those who joined late, can you share a little bit again about where this system can be installed, whether it's at the end of a loop?
I'll go back to that slide. So here's the places that we would normally look at is depending upon a hot water system or whatever. This is showing a hot water WFI system. We would install a unit directly after the pump, downstream of the pump, because that's where the most amount of Rouge actually develops inside the piping system. And in order to look at the the piping on the return line, much like what we do with the TOC analyzer, in order to look at the worst case scenario of how the water, the temperature, the chemistry is interacting with the piping at that particular time. So we would have one directly after the pump, one on the return line, very close to the storage tank on the return line.
Absolutely. And the very last question for today, and again, for any questions we didn't get to today, we'll be sending out a list of questions with corresponding answer with more information after the webinar. And the question is, the system monitors Rouge rate and accumulation. Do you recommend the monitoring of any other parameters to ensure that we get the full picture of the system?
Rouge is dependent upon temperature. Rouge is dependent upon your operational characteristics of your water system and your points of use, whether they're open or not. When the water is recirculating with no use points open, the Rouge will develop greater inside the piping. So understanding these dynamics and having the data in order to show what's going on with the water chemistry at any given moment, and the temperature is going to be indicative of what happens with the Rouge rate. It's all interactive. So having knowledge of what's going on and having that data time-stamped inside your computer system, where you can have your Rouge data, your temperature data, your flow rates, and what is operational or not operational at your point of use, is all going to be dynamically intrusive to how the Rouge develops over time.
Absolutely. Thanks very much, Nissan. And for anyone, again, we'll be answering any questions that we missed today in the follow-up email. And we'll also share Nissan's contact information as well. If you have any other questions, feel free to reach out. We'll be happy to answer those. Thank you, everyone, for joining us today. Again, we really appreciate you taking the time, and I hope everyone have a very good day.